Welcome to this explainer. Today, we're going on a bit of a microbial manhunt. We're bringing some serious energy to the lab bench because we are decoding the identification traits of a truly notorious bacterial family, Streptococcus. Our mission? To figure out exactly how scientists track down, profile, and ultimately catch these microscopic suspects that cause massive headaches in veterinary medicine. To kick things off, I want you to ask yourself this. What do a highly contagious feverish horse and a dairy cow with infected milk actually have in common? On the surface, well, absolutely nothing. But if we look under the microscope, it's a completely different story. Today, we're diving into a microscopic detective story, tracing real laboratory clues to solve some serious, real-world veterinary cases. Okay, let's just lay out our structural roadmap for the investigation ahead. We'll start by profiling our microscopic suspect, unpack the lab detective's toolkit, dive into our two case files, bovine mastitis and equine strangles, and finally, we'll compare our culprits side by side. Step 1. Profiling the microscopic suspect. Now, what's really fascinating here is how a simple negative enzyme test and a basic physical shape give us our very first forensic clue to separate two famous bacterial cousins. Under a Gram-stain, both Streptococcus and Staphylococcus show up as gram-positive spheres, or what we call coccus. But check out the difference. Streptococcus divides in a single plane, so it forms these distinct long chains. Staphylococcus, on the other hand, forms these cluster-like shapes. But the ultimate separator? The catalase test. If you drop some hydrogen peroxide on them, Staphylococcus bubbles up immediately. It's catalase positive. Streptococcus? It does absolutely nothing. Strictly catalase negative. So, let's flesh out this profile a bit more. Beyond being gram-positive spheres and chains, these suspects are non-modal, meaning they can't just swim away, and they don't form tough spores. They're facultatively anaerobic, so they can survive perfectly fine with or without oxygen. But here's their Achilles heel, a critical vulnerability. They are highly susceptible to desiccation, basically drying out. Now, as a lab detective, this tells you something huge. It means clinical specimens, like a swab from an infected animal, have to be collected and transported with incredible care. If you don't, your evidence literally dies before it even reaches the lab. Moving on to part 2, let's head into the interrogation room and look at the lab detective's toolkit. Our first major tool is haemolysis. You can think of this as the pathogen's modus operandi when it's placed on a blood agar plate. When we grow Streptococcus on this agar, we watch exactly how it destroys the red blood cells. Alpha hemolysis is a partial destruction, which leaves this greenish discoloration. Beta hemolysis, that's complete destruction, leaving a totally clear zone right around the colony. And gamma means there's no visible effect at all. Those beta hemolytic strains, those are generally your major red flags. They tend to be the most pathogenic and, frankly, the most dangerous of the bunch. Next up, we use something called Lancefield grouping. Think of this as our serological fingerprinting technique. It's a brilliant system for unmasking different species based on these specific carbohydrate antigens found in their cell walls. We basically extract these antigens and use latex agglutination. This is where antibodies bound to latex beads will actually clump together if they find their matching antigen. It's like a lock and key. This lets us sort our suspects into groups A, B, C, D, and so on, giving us a massive shortcut to identifying the exact species we're dealing with. But these suspects don't just go down without a fight. They possess a serious arsenal of virulence factors to evade the host's immune system. They deploy enzymes like streptokinase to dissolve clots, hyaluronidase to break down connective tissue, and DNase to destroy DNA so they can spread further. But even more devious are their antiphagocytic shields. They have this thick polysaccharide capsule and a specialized M protein on their cell wall. These structures act as molecular armor, actively preventing the host's immune cells, like macrophages, from swallowing and destroying the bacteria. Let's put these tools to work. Opening case file number 3, bovine mastitis. Okay, here is the actual sequence of our laboratory workflow, showing how clinical suspicion translates into hard evidence. First, aseptic milk collection is absolutely vital. We cannot have environmental contamination ruining our crime scene. Step 2, we culture the milk on blood, Edwards, and McConkey agars at 37 degrees Celsius. Step 3, we get under the microscope to confirm those gram-positive chains. And finally, step 4, we run our specific identification tests to nail down exactly which Streptococcus is causing the farm's outbreak. And speaking of those identification tests, enter the CAMP test. This is our absolute smoking gun for this case. Named after Christie, Atkins, and Munch Pedersen, the test is just beautifully elegant. we streak our unknown Streptococcus perpendicular to a known line of Staphylococcus aureus. If our suspect is Streptococcus agalactiae, it actually produces a specific factor that interacts with the Staph aureus, creating this highly distinct arrowhead-shaped zone of complete beta hemolysis. If you see that arrowhead, boom, you've caught S. agalactiae red-handed. So the crucial point is right here on this lineup when we evaluate our three primary bovine suspects. S. agalactiae lives right in the milk ducts, is Lancefield group B, and as we literally just saw, is camp positive. S. dysgalactiae hangs out in the environment, is group C, but is camp negative. And then we have S. uberis. This is an environmental opportunist that isn't assigned a Lancefield group, is camp negative, but tests positive for esculin hydrolysis. By simply running a camp test and an esculin test, we rapidly eliminate suspects and pinpoint the exact ecological source of the outbreak. All right, let's move to our second investigation. Section 4, Case File Equine Strangles. Our main culprit here is Streptococcus equi subspecies equi, and its profile is striking. It's a Lancefield group C and intensely beta hemolytic. On an agar plate, it forms these large mucoid colonies that can be up to 4 millimeters across, which are massive red flags for severe respiratory infection. Clinically, it causes strangles. This is a highly contagious disease in horses characterized by high fevers and severe abscessation of the regional lymph nodes. And it spreads like wildfire through purulent exudates when horses mix, at sales or shows. But here's the tricky epidemiological twist that makes strangles such a nightmare to control. Long after a horse appears to have fully recovered, the bacteria can actually hide. These chronic convalescent horses can asymptomatically harbor S. equi in their guttural pouch, which is a sac in the back of a horse's throat. They act as hidden, silent reservoirs, shedding the bacteria and starting whole new outbreaks. Because of this, highly sensitive PCR testing is absolutely essential to track down these hidden carriers before they can infect a new herd. I really want to inject a surprising and honestly pretty serious fact right here. While 99% of strangles cases are typical, meaning they're restricted to the upper respiratory tract and local lymph nodes, about 1% of cases develop into what is aptly named bastard strangles. In this devastating complication, the bacteria break out of the local nodes and spread systemically, causing abscesses in multiple internal organs. It perfectly illustrates the incredibly high stakes of this infection and just how destructive this pathogen's virulence factors really are. And this brilliantly illustrates exactly how we definitively close the equine case. You see, S. equi has some incredibly similar relatives, like S. zooepidermicus and S. equisimilis, which are also group C Streptococcus. To separate them, we use specific sugar fermentation profiles. Notice how S. equi is kind of the lazy one here. It ferments almost none of these sugars. It's negative for sorbitol, lactose, and trehalose. Zooepidermicus, on the other hand, ferments sorbitol and lactose. So by feeding them different sugars and seeing what they digest, we get our final undeniable identification. Bringing it all together now, Section 5, Comparing the Culprits. I want you to notice how the exact same foundational traits, gram-positive spheres and chains with a negative catalase test, branch off into two completely different ecological and diagnostic profiles. In our bovine case, we're dealing with a trio of suspects causing mammary gland infections, and we solve it using camp and esculin tests to trace whether the spread is environmental or from the milk ducts. Over in the equine case, we have a single, highly contagious culprit causing severe lymph node abscessation, and we solve that one using sugar fermentation and PCR to hunt down asymptomatic carriers. Which leaves us with this final thought. It's truly remarkable, isn't it? How does a simple microscopic shape, tiny spheres in a chain, combined with a single negative enzyme test, hold the absolute power to unravel such complex, devastating, real-world disease outbreaks? The next time you hear about a farm quarantine, just remember, the biggest mysteries are so often solved by the smallest, most invisible clues. Keep exploring, keep learning, and I'll catch you on the next explainer.